So we're going to begin today just by reviewing our oxygen hemoglobin dissociation curves, because it feels like it's been a little while and that's a pretty chunky topic. So I'm going to draw along with you, but you may try to get a little bit ahead of me. So when we're talking about our hemoglobin oxygen dissociation curves, we're talking about this curve that describes the saturation of our hemoglobin with oxygen versus our CO2. Okay. So our usual curve looks like this. Then when we look at it, we think about right shifts and we think about left shifts. So our right shifts and our left shifts have to do with changing the affinity of hemoglobin for oxygen. So basically, if we move something to the right, we move something to the right, what we're saying that we now need a higher partial pressure of oxygen to get the same saturation. We have lowered affinity versus a shift to the left at a lower PO2. We get that same saturation, meaning we have higher affinity. So a lot of the changes to the affinity of hemoglobin for oxygen, we've talked about pretty much all of them, can be understood as responses to active tissue in order to get it more oxygen. So what we want to do in order to get our active muscle more oxygen is we want to make it easier to unload oxygen from hemoglobin. So we want to get it easier to get those oxygen out of their fourth loss in hemoglobin and into our tissue. So we're talking about right shifts happening at the tissue. And sometimes we have left shifts happening up at the lung edge, but focusing on what's happening at the active muscle is really the easiest way to think about it. So we have a number of factors that affect our hemoglobin oxygen dissociation curve. So one is temperature. And these are all going to increase our unloading. So they're all going to be right shifts. So we'd have temperature. So in active muscle, as you're moving it, you're giving off heat as a byproduct. So increases in temperature are one of the ways that we are helping to unload oxygen. We also have changes to pH. So pH gets more acidic in active tissue. We have acid buildup. So we have a drop in pH, also creates this right shift. We have an increase in carbon dioxide as we're creating more ATP. And as we work harder and longer, if we create a low oxygen condition as we use up the oxygen, we'd also have an increase in our 2,3-BP2. So this is what I think is the simplest way of thinking about which direction these shifts are happening and why, is to think about what would happen to the muscle as it's active and realize that if it's happening in the muscle while it's active, then that's probably going to shift us right, decrease affinity of hemoglobin for oxygen, and therefore help us get more oxygen into the tissue. So then if you ever need to think about a left shift, you just think backwards, basically. So the next thing we want to think about is carbon dioxide transport. So the role of breathing and respiration, right? We're trying to get oxygen from our environment into our lungs and ultimately into our tissue in order to fuel the creation of ATP. But we also have to get rid of carbon dioxide. We'll see that blood pH is really important. It is one of our homeostatically controlled variables. So it's important that we don't let the blood get too acidic. And carbon dioxide actually acts as an acid in your body. So it's really important to breathe off carbon dioxide. So this is going to happen. Carbon dioxide exchange will happen in the systemic capillaries, so by the tissue, and also in the pulmonary capillaries in the lungs. So we're getting carbon dioxide out of our tissue into the blood, and we're getting it out of the blood into those pulmonary capillaries, into the alveoli, and breathing it out. So we've seen that carbon dioxide affects the transport of oxygen, and we're going to see that the opposite is also true. Oxygen affects the transport of carbon dioxide. But first, let's take a look at where we have our carbon dioxide. So we're listing a number of different kind of types of carbon dioxide here. So first, we have our carbon dioxide that's just straight up dissolved in the blood, right? So carbon dioxide just dissolved directly in the blood is around 5.5%, right? Slightly higher in the venous blood because we picked up carbon dioxide. We also talked about, when we were talking about the Carbamino effect, that we can bind carbon dioxide to hemoglobin, right? And we know that the Carbamino effect, when we have hemoglobin bound to carbon dioxide, that decreases hemoglobin's ability to carry oxygen. So we know there's got to be some on there. In arterial blood, about 5%, again, a bit higher in our venous blood because we've increased the amount of carbon dioxide there. But we'll also notice that we have dissolved as bicarbonate on this list. So when we think about carbon dioxide, we're going to have to think about the fact that carbon dioxide actually breaks up. Right? We can break it up into hydrogen ions and bicarbonate ions. So most of the carbon dioxide that's moving around in your blood is actually in this form. So the way we would appreciate the carbon dioxide, see the carbon dioxide, measure the carbon dioxide is by the amount of bicarbonate in the blood. We can see that's like 90% in the arteries and only slightly lower in the veins. See carbon dioxide in all of these places, but mostly we think about these ions when we think about carbon dioxide. Yeah, no problem. Yeah, somewhere on the globin part. I don't even know how to name the globin parts, but they have names. But yeah, not to the heme groups, not to the iron, just kind of separately on the polypeptide chain. So here's what's going on with that bicarbonate bit. So we mentioned very briefly when I talked about erythrocytes that they have this enzyme carbonic anhydrase in them. So this is an enzyme that can take carbon dioxide and turn it into carbonic acid. That's why it's called carbonic anhydrase. Right, so we start with some carbon dioxide. We need to add water, but there's plenty of water in our blood. Right? We know our plasma is mostly water. So with carbonic anhydrase, we create our carbonic acid. This is a reversible reaction. So we can take carbon dioxide, turn it into carbon dioxide, carbonic acid, or we could take carbonic acid and turn it back into carbon dioxide. That's going to be important when we get to the lungs and we think about, okay, how are we going to breathe off bicarbonate ions? Okay. But for our purposes now, what we need to also realize is that carbonic acid, we're not like totally done. The carbonic acid also separates up into our hydrogen ions the amount of hydrogen ions is basically how we get pH, right? That's what the H is. So acidity is hydrogen ions. And then we have bicarbonate ions as well. Okay, so it's this sort of breakup of carbon dioxide. So turning the carbon dioxide into the carbonic acid and then into these ions that makes carbon dioxide an acid for our body. Again, looking at these equations, we've seen the law of mass action before, but all that means is if we have an increase on one side of the equation, it's going to cause more of that direction to happen. So if we have more carbon dioxide, we're going to create more carbonic acid and therefore more bicarbonate and hydrogen ions. Or if we were to have tons of bicarbonate, we might go the opposite direction, turn it into carbonic acid, and then back into carbon dioxide. So when we're looking at the relationship between carbon dioxide and partial pressure of carbon dioxide, so the total amount is here on the y-axis, and then our partial pressure is here on the x-axis. So taking any one of these curves, we could see that as our partial pressure of carbon dioxide goes up, so does our total blood carbon dioxide, right? So this is kind of an indirect measure of the amount of carbon dioxide in our blood. But we can also see that we have 3 different colored lines, which usually means we're talking about a shift in our book, right? So what we're looking at here is changes in the partial pressure of oxygen. So changes in the partial pressure of oxygen. And I would also note that up at the top, we start with 0 partial pressure. And then as we're moving down, we're increasing the partial pressure of oxygen. That's what's happening here. So just key in on that. Because what we're going to notice here is that basically, if your blood has higher partial pressure of oxygen, more oxygen in it, it becomes harder to carry carbon dioxide. So that's why we're lower here. Whereas if there's not a lot of oxygen, it becomes easier to carry carbon dioxide. There's more of it. So there's this trade-off between oxygen and carbon dioxide, which probably doesn't come as a surprise, given we've been tracking how we're offloading oxygen and then loading on carbon dioxide as we move from the arteries to the veins. So if we're thinking about what's happening at the blood and at the tissue, we may want to think kind of globally about what's going on with both oxygen and carbon dioxide, because this is a lot of different things affecting each other. So if we're starting up here we're talking about respiring tissue. So one thing that's happening is that our partial pressure of oxygen in the respiring tissue is low, which means the partial pressure of oxygen in our blood is dropping, because directly tied to, we're unloading that oxygen into the tissue, right? So that's a direct kind of connection between the dropping partial pressure of oxygen and the O2 unloading. But once we start to consider the carbon dioxide, there's a lot more going on. So we have 2 reasons at the tissue that we are increasing the carbon dioxide that's getting into our blood. Okay. One is that trade-off that I just mentioned between partial pressures of carbon dioxide and partial pressures of oxygen. All right, so as this partial pressure of oxygen is dropping, that makes it easier to carry carbon dioxide. We get more carbon dioxide loading into the blood. Another thing, a direct effect, is just when we have more carbon dioxide here in the tissue, we're loading more carbon dioxide into our bloodstream. Next, we have our kind of indirect effects. So one of these is the Carb-Amino effect, that right shift in our hemoglobin oxygen dissociation curve. So we have more carbon dioxide, which means we've lowered hemoglobin's affinity for oxygen. Right, so we put carbon dioxide onto our hemoglobin. It's harder to carry oxygen now, so oxygen breaks off, increasing the oxygen unloading. And at the same time, some of that buildup of carbon dioxide is turning into carbonic acid, which is turning into our hydrogen ions and our bicarbonate ions. And we know that pH, a low pH is an increase in hydrogen ions. So low pH is a lot of hydrogen ions. Either of those tells us we have acid. So we have an acid buildup. This is our Bohr effect. So this is also creating a right shift, a decrease in that affinity for oxygen. So all of this is kind of working together at the tissue end in order to make it easier to unload oxygen. And also, if we're thinking about the carbon dioxide, which we might want to when we're thinking about breathing carbon dioxide off, we're also going to talk a little bit about chemoreceptors and how important it is to breathe off carbon dioxide. So both the partial pressures of oxygen and carbon dioxide are also helping us load more carbon dioxide into the blood. So these are kind of our two goals at the tissue. So now we're at the lung end. So at the lung end are two goals, right? We still have goals. The one that's highlighted on here is that in the lungs, we want to put more O2 onto the hemoglobin because we want to freshly oxygenate our blood, get more oxygen to the tissue. Another goal of ours is to get carbon dioxide out of the blood so that we can have it diffuse out of those capillaries into the alveoli so we breathe it out. Also important. So this again does partially go back to those partial pressures of oxygen and carbon dioxide in the blood. So direct effect of oxygen, we have more oxygen, so it's going on to the hemoglobin. We're increasing oxygen loading. So that part is fairly straightforward. Another effect of that increase in carbon dioxide, that increase in oxygen, is that we have this trade-off between oxygen and carbon dioxide. So we have low carbon dioxide, and we're increasing carbon dioxide unloaded. So functionally, we're decreasing the carbon dioxide. Then we have our other effects. So this lowered carbon dioxide, increasing the carbon dioxide unloading. And this lowered acidity is improving, this time, hemoglobin's affinity for oxygen. So this is the opposite of what we've had happen in the tissue. So this is actually going to be a left shift. So this is going to increase our oxygen loading, getting more oxygen into the blood. So hopefully on Friday or over the weekend, you took a look at the regulation of ventilation, this first section. Gave you this first part because while it's cool, you won't be extensively tested on the part involving the nerves. At most, I'd expect to see like one question. But just running through quick in case you missed it. Central regulation of ventilation, we're controlled by neurons and respiratory centers in the brainstem. We're controlling our skeletal muscles using motor neurons. We can look at differences in our action potentials depending on quiet versus active breathing, and we see that we have kind of alternating things going on between our different types of intercostals. We have both our peripheral and central nervous systems involved. So both things that are not part of the brain and things that are part of the brain. We looked at a couple respiratory centers in the brainstem. And we talked about some different groups here, inspiratory neurons versus expiratory neurons and some of their roles. More action potentials related to inspiration and expiration. Talked a little bit more specifically about the pons. Backwards. And we know that there's theoretically a central pattern generator, but we don't know where it is or what it does. So what we're going to focus on today is chemoreceptors, sensing changes in your blood and in CSF, and how this affects changes in respiration. So we have a number of signals that are coming in to our brainstem to talk to us about what our respiration rate should be. Two of the most important ones are our central chemoreceptors and our peripheral chemoreceptors. These are really telling us about the contents of the blood. We do have other sensory input, so we can sense the stretch in our lungs. We have stretch receptors. We can sense irritants, so we can sense like smoke. We can sense how our body feels, so proprioceptors. So all sorts of stuff is going in there as well. But ultimately, we're thinking about how we set our breathing rhythm. We'll do review questions on Wednesday. Okay. So thinking about the chemoreceptors, pulmonary stretch receptors, irritant receptors, and proprioceptors in the muscles and joints that are telling us the position of the body. Focusing in as our main thing we want to understand are the chemoreceptors. So chemoreceptors, depending on which ones we're talking about, can sense a couple different things. As a group, they're detecting blood levels of oxygen and carbon dioxide, but this may not be directly, and it may not be every chemoreceptor, and we'll talk about the differences. But first, we're going to think about where they are. So one type of chemoreceptor we're going to see is going to be in the carotid body. So I think you saw or will see in lab where your carotid artery splits into internal and external. You saw that carotid sinus. At that split, we have the carotid body. And it's really important, and we talk about it when we talk about that split, because it's one of the things that's telling your brain, okay, do you have enough oxygen? Okay, you have too much carbon dioxide. It's great that it's located right there, right? Because that means it's going to regulate the contents of those things going into your brain, right? Because we're right there at the juncture of the two carotids. The other place that we have a chemoreceptor is in the brainstem itself, in the medulla oblongata. Okay, so that kind of thinner part inferior to the pons. So we consider that our central chemoreceptor. So peripheral, right, is anything that's not part of the brain or the spinal cord. So the carotid body, not part of the brain or the spinal cord, so it's peripheral. Medulla oblongata, part of the brainstem, so that's our central chemoreceptor. So we're going to start with that peripheral chemoreceptor in the carotid body. So because it's right there at the juncture of the internal and external carotid body, carotid arteries, it's in direct contact with the blood. So it's going to communicate with our neurons, afferent neuron, afferent going up into the brain, into those respiratory centers to give us information, using chemical messengers. So they're going up to the medullary respiratory control areas. What they're responding to mainly changes in blood pH. So this is where it's important to remember that carbon dioxide can turn into carbonic acid, can turn into hydrogen ions and bicarbonate ions. So by measuring blood pH, this is an indirect kind of measure of carbon dioxide. Like this, we have as carbon dioxide rises, it pushes that equation to the right, creating more hydrogen ions ultimately. Our peripheral chemoreceptors do also respond to O2 changes, but they have to be pretty drastic. It's actually mainly the amount of carbon dioxide in your blood that changes your respiratory rate. So here we see the effects of partial pressure of oxygen on ventilation. So it actually makes most sense to start over here at the right side of the graph. So this is our normal resting level. So normally our arterial partial pressure of oxygen here, 100. Okay. And here on the left, looking at minute ventilation, which our tidal volume by our respiration rate, right? So the way we're going to change this is by changing that respiration rate, breathing faster or breathing slower. So as we start to have partial pressure of oxygen dropping, it takes until we get to about 60 before we start to see significant changes to our respiration rate. So oxygen can fall pretty far in arterial blood before we start to change our respiration rate significantly. It's very low. Here we see the converse kind of curve for carbon dioxide. Our normal resting level, our normal amount of arterial carbon dioxide is 40. Okay. 46 in venous blood, 40 in arterial blood. So we were here at that same point for our minute ventilation, okay, which is our tidal volume, our respiration rate. We can see that instead of having that long kind of tail that we were following with oxygen, something like that, we don't have the mirror image of that for carbon dioxide, right? A mirror image would look something like that, right? If we had this slow change. But instead what we see is there is an immediate kind of shot up as arterial carbon dioxide levels rise. So this is why we're responding more to carbon dioxide than oxygen levels, because as soon as we have these carbon dioxide levels change, we start to change the respiration rate. Now, you might notice is that there's a point at which it peaks. Okay. So here we have a partial pressure of carbon dioxide of about 90. The reason we have a peak and a fall here is because you go into a coma if your carbon dioxide levels get this high. Being in a coma messes up your respiration rate. Okay. So that's why we start to see this decrease in minute ventilation happening. If you're in a coma, not doing great. You can survive a partial pressure of carbon dioxide of 90. If you get to above about 180, that calls it death. So carbon dioxide can get so high that you die. You would notice before you get there, though. So now we're going to think about the central chemoreceptors in particular. So the carotid bodies in the peripheral chemoreceptors, we're sensing both oxygen and carbon dioxide levels. We're doing both. Partially, they were sensing carbon dioxide through the concentration of hydrogen ions. In our central chemoreceptors, now that we're on the medulla, now that we're in the brainstem, we have a blood-brain barrier. So we're not like directly in contact with the blood. So the central chemoreceptors are actually responding to changes in cerebrospinal fluid. So CSF, that fluid that's bathing surrounding the brain. Now, we don't directly respond to CO2 if we're essential chemoreceptor. What's happening is that there's going to be an indirect relationship between CO2. It's going to be mediated by the pH. Right? So we're not directly just shoving a bunch of carbon dioxide into the cerebrospinal fluid. What's happening is that an increase in carbon dioxide means we turn it into carbonic acid, so we make more carbonic acid, which then dissociates into the hydrogen ions and the bicarbonate ions. And it's those hydrogen ions that cross through to the cerebrospinal fluid and those central chemoreceptors. It's also helpful to know that if we have hydrogen ions and bicarbonate ions, they can snap back together to form the carbonic acid and then give us carbon dioxide again. So the central chemoreceptors are indirectly responding to carbon dioxide levels because they're responding to pH. And they're not actually responding to oxygen levels at all. So it's just the blood oxygen going up to the brain. Once you're in the brain, hope you got the oxygen. They're not checking. So our key number to remember for partial pressure of oxygen, 60, related to these curves. All right, so here we just see again that slow response to dropping oxygen levels, but this faster response to rising carbon dioxide. And in our next unit, we're going to talk about the regulation of blood pH again. So we're going to still be thinking about respiration because we're going to be thinking about carbon dioxide and how it is an acid, creates hydrogen ions and bicarbonate ions. But we're going to add in the fact that the kidneys have effects on this as well. So here we see a picture of that central chemoreceptor. So this is potentially helpful for understanding what's going on with that kind of indirect effect. Okay. So our blood-brain barrier here between the CSF and our capillaries here. We have nice tight junctions here between our capillary endothelial cells. We have assisting glial cells that are not letting things cross directly in much. What we have happening is that we have carbon dioxide and water here forming carbonic acid, and then we have our hydrogen ions getting to the central chemoreceptors, basically. So here we see another big maze of what's going on here. So we're going to automatically respond to these changes in oxygen levels, but especially carbon dioxide levels. So we refer to these changes in ventilation and in respiratory rate as chemoreceptor reflexes. Reflex, not like you're thinking, oh gosh, I need to breathe off all this carbon dioxide. It's just happening. So if our arterial blood, if our arterial blood drops low enough in oxygen concentration, then our peripheral chemoreceptors can detect and respond, helping us to breathe faster, so increasing ventilation to get more O2. But what we more likely have happening are changes with our carbon dioxide. So our carbon dioxide, as it builds up in arterial blood, our peripheral chemoreceptors can directly respond to that, again, increasing ventilation. Or because carbon dioxide ultimately creates hydrogen ions, these hydrogen ions are also detected by the peripheral chemoreceptors, increasing ventilation. And we also have our central chemoreceptors. So as we have increases in carbon dioxide, this increases hydrogen ions in the CSF. And then the central chemoreceptors respond only to this pathway, right? Only to build up in carbon dioxide, creating acidity, so increase in hydrogen ions, increasing ventilation. But that's okay. It's like a backup system, right? Both systems are working at once. It's not like you have one or the other working. So here we see different breathing rates and how they're related to these chemoreceptors and to our partial pressures of carbon dioxide especially, but also oxygen and hydrogen ions. So hypoventilation. Right, so if you are hypoventilating, you're breathing too slowly, you're not getting enough oxygen. You're not getting enough oxygen, but even more importantly, you're not breathing off enough carbon dioxide. So we start to see low partial pressures for O2 in arterial blood, high partial pressures for carbon dioxide. And because we have high partial pressures of carbon dioxide, we also have high levels of hydrogen ions, which means a low pH or highly acidic. Our chemoreceptors, both the peripheral and the central ones, are responding to a grab bag of these factors, depending on which chemoreceptor we're talking about. Peripheral, sensing all of them. Our central, sensing hydrogen ions, which is an indirect measure of carbon dioxide. So we've been breathing too slow, we've been hypoventilating. So now we're going to increase ventilation, breathe faster until we correct our partial pressures of carbon dioxide and oxygen. We want oxygen to be at 100. We want carbon dioxide to be at 40 in our arterial blood. Once we're back there, we go to our sort of baseline ventilation rate. In hyperventilation, this may or may not be incorrect on your slide, but I think I found an error reviewing these. So in hyperventilation, what's happening is you're breathing too fast. Right? You're breathing too fast, which actually means you're breathing out a ton of carbon dioxide. And you actually can have too much oxygen, right? Too much oxygen, you're getting lightheaded. At the same time, your blood is dropping the concentration of hydrogen ions, which means its pH is rising, it's becoming more basic. Chemoreceptors sense that you're doing this. They sense that you're breathing too much, you're breathing too quickly. The respiratory control sensors are going to send a signal out telling you to decrease your ventilation rate. Decrease your ventilation rate until you correct, get back to our normal partial pressures, and then you're back to your normal breathing rate. Our final concept, really briefly, is talking about ventilation and perfusion ratios. So when we say ventilation, we're thinking about the flow of air. When we talk about perfusion, we're thinking about the flow of blood. And we can compare these two things. So we create a ratio matching how much ventilation we have happening. These are rates, so usually I think we see little dots over them. But all we're saying is, okay, so we're dividing our rate of airflow by the rate of blood flow. So ideally, we want them to be the same, okay? So our normal situation is that our rate of air going in to the alveoli is pretty much the same rate as the blood flowing past them in the pulmonary capillaries. So what we're checking to figure out what kind of blockage we may have is whether we are above or below 1. right? So we're looking at changes in this ratio. So I'm going to actually write this out on the side because having everything smushed together, I think, makes it a little more difficult. Okay, so what we're saying in this first one is that if we obstruct the airway, we close it off, I don't know, you have something stuck in your throat, you really can't breathe, we don't have as much airflow, right? So we're dropping down our ventilation. So is this going to give us a ventilation perfusion ratio above 1 or below 1? You can just do it with your thumb. Go below, right? So we've dropped this number, we're dividing by the same number, so we're below 1. So this is a way we can tell we have an obstruction here. The opposite is going to be true if we occlude the blood vessel. So if we have some kind of a clot, right, we're going to see that our ventilation is staying the same, but our perfusion is dropping down. So we're going to see a number below 1. Sorry, above 1. So we care about this because we're kind of circling back to a concept we talked about a little while ago, which is that we do have bronchodilation and bronchoconstriction happening, changing the radii of our airways. And also, as we learned last unit, we have vasoconstriction and vasodilation happening, changing the radii of our blood vessels. So when we have a change in this ratio, this rate of ventilation versus perfusion, because this changes the buildup of carbon dioxide or oxygen in the bronchioles and in the arterioles, it's going to have an effect on the relative dilation or constriction of these vessels. So if ventilation is decreasing, we're going to have an increase in partial pressure of carbon dioxide, and we're going to have a decreased partial pressure of oxygen in both the blood and the air. And that partial pressure of carbon dioxide, the smooth muscle in the bronchioles responds to that with dilation. So they're increasing in size. And the arterioles passing by are going to respond to the fact that their oxygen is dropping. And they're going to respond here with vasoconstriction. Now, if we change the perfusion, we also have similar things happening. This is just that we're kind of seeing the opposite reason. So we have perfusion decreasing. What we're saying is the blood flow is decreasing. So our blood is moving kind of slowly. Maybe it's picking up extra carbon dioxide. Maybe it's giving off, picking up extra oxygen, giving off more carbon dioxide. So then our arterioles have more oxygen in them and they respond with vasodilation. The bronchioles sense this lowered partial pressure of carbon dioxide and respond with bronchoconstriction. And then we just have a figure of this. Yeah, and as you're thinking about this, I do recommend that you think about these things more in context of like the last time we talked about bronchodilation and bronchoconstriction. If there's any question about ventilation perfusion ratios, again, that'll just be like one tops. All right, so that brings us to the end. The rest is just going through this figure, which is kind of telling you how this ventilation and perfusion is working. But we now know everything there is to know about a lung. And everything there is to know about the blood. Can that possibly be true? It's not true. But I wish. Close enough.